and good afternoon, everybody. So my aim today is just to give you a very high-level overview of how I came to be at NINR, my current role here, and my future career plans. In particular, I will discuss the collaborative framework that we have developed within the Neuromuscular Symptoms Unit. I'll give you a brief overview of the patient population with who we work, and my role both in translational research and in clinical trials. So I completed my bachelor's in sports science, focusing in nutrition between 2010 and 2013 at the University of the West of England, which is in Bristol in the UK. And this is where I really developed a passion for clinical research. As part of my undergraduate dissertation project, I looked at the effects of hyperhydrating solutions on endurance performance of elite oarsmen. I then was awarded a scholarship to conduct a PhD at the Nutrition Innovation Centre for Food and Health, which is based at Ulster University in Coleraine, right at the northern tip of Northern Ireland. And as part of that, I completed 2 observational studies and 2 clinical trials. The first clinical trial was a parallel group randomized placebo-controlled trial, looking at vitamin D administration versus placebo on health and performance outcomes in Irish footballers. And the second trial was a comparative efficacy trial, looking at the relative bioequivalence of vitamin D administered via oral spray solution, which bypasses the GI tract, versus capsules, which is the typical practice. I finished my PhD in early 2016, and I had some time on my hands, so I joined my wife's lab at Queen's University Belfast Centre for Experimental Medicine, where I gained additional experience in cell culture techniques, particularly looking at oxidative stress of the retinal pigment epithelium. And we both applied for postdoctoral positions at NIH, and here I am. And here she is. So why did I come to NINR? Well, a major reason is because I had gained all this experience working with elite-level athletes. This is not typically considered, under most circumstances, a patient population, and I wanted to really translate the skills that I had learned in clinical trials to a patient population with unmet medical need. And I felt that Dr. Mayer's research program really encompassed this because we focus on a rare neuromuscular disease called RYR1-related myopathy. And just a brief blurb about rare diseases. So orphan designated diseases, as the agency FDA refers to them, or rare diseases affect less than 1 in 200,000 individuals in the United States at any one time. And for these rare diseases, there's very little financial incentive for big pharma to invest in developing drugs. And I felt that this is where NIH can really step in. We can actually tip the balance toward conducting these early phase clinical trials, which provides the groundwork for phase 2 trials later down the line. And this statistic I really love on the right-hand side. When we consider rare diseases individually, they are rare, but collectively this is simply not the case. And in fact, 1 in 10 Americans will be diagnosed with a rare disease in their lifetime. So here's the collaborative framework which Dr. Mayer and her team have managed to establish. So we partner with the pharmaceutical industry, which is essential in terms of bringing new drugs to market. We work with a wide range of intramural institutes and centers, as well as academic institutions. So onto the patient population. So RYR1 is a large gene, it's 106 exons, and this encodes a transmembrane protein, the largest ion channel in humans, which is responsible for releasing calcium from the sarcoplasmic reticulum, which is a specialized form of ER within muscle, into the cytosolic space. This is essential for muscle contraction, otherwise known as excitation-contraction coupling. Now, the incidence of RYR1-related disorders, which stem from variants in the gene I just mentioned, are estimated to affect around 1 in 90,000 individuals in the US. However, this is likely an underestimate. And in the past, this range of heterogeneous disorders have been defined based on histopathological findings, which we now know are non-specific. So for example, if I can just draw your attention to the bottom left, you have an NADH stain, which stands for oxidative enzyme activity. And you'll see that there are amorphous cores, or areas absent of staining in the center of each fiber. And this was first described in the early 1960s by McGee and colleagues as central core disease. Now, what we're understanding is that these features are not necessarily static and they can change over time. And so this nuanced nomenclature is something we're looking to address as a team. And here's the protein. So, as I mentioned, it's embedded within the SR membrane, and it encodes the largest ion channel in humans. Variants in the gene RYR1 can result in defective intracellular calcium flux. So that's either increased calcium release or channel leakiness, decreased calcium permeation into the cytosol, or actually decreased expression of the protein itself. And we focus a lot as a team on symptoms and the symptom science model, and this is really an ideal disorder in which to embody that because it's highly heterogeneous. So what we do know is that recessive cases, or cases with a recessive mode of inheritance, are typically associated with a more severe clinical phenotype and can range from delayed motor milestones, contractures, proximal muscle weakness, and scoliosis, and in the more severe cases, respiratory muscle weakness and ophthalmoplegia. An important side note actually is that this disease is considered allelic or directly linked to malignant hypothermia susceptibility, which is a hypermetabolic, potentially fatal condition, which is most well known for its attribution to inhaled anesthetics and muscle relaxants, but more recently has also been linked to exercise in the heat and viral exposure. So what we have here at the bottom is a number of images. And when you look at this initially, you may think that these individuals have different diseases. For example, on the right-hand side, under John Bluth and colleagues in 2005 and 2007, at first glance, look relatively less severe than under above Clark and colleagues in 2010. However, these individuals all have an RYR1-related disorder. And so there's a lot of overlap between both clinical phenotype, that's all linked to variants in the RYR1 gene. So now I'm going to briefly talk to you about my work, which I categorize into translational and preclinical, and then clinical trials. So our team co-wrote a grant application for the Directors Innovation Award back in 2017, and we were successfully awarded this for transforming orphan drug research via translational science collaborations. And the aim of that grant was really to foster intramural collaborations. So you see here we work with a number of different institutes, including NIDA, drug abuse, NCAT, Center for Advancing Translational Sciences, Neurological Disorders and Strokes, and Child Health Development. And the aim of this project was really to identify compounds that were hits on a high throughput screen as being able to stabilize this channel, which was on the previous slide, and then to assess that in vitro models of the disease, and then in an in vivo zebrafish model of the disease. On the right-hand side, in black, you have skills that I came in with, and in red, you have skills that I've been able to develop through the support of the NINR Intramural Program. On the top left-hand side, you have a wild-type zebrafish, and on the right-hand side, you have a mutant zebrafish. You'll notice the slightly crooked phenotype as it's moving now on the right-hand side. So this is the zebrafish model which we are trying to improve swim distance in with the addition of the small molecules which are identifying through our high-throughput screen. And we have actually managed to identify a particular compound that's demonstrated early-stage efficacy in terms of swim distance, which you see in the bottom center box plots there. So in terms of preclinical research, as was mentioned earlier, Dr. Mayer recently completed the first natural history study and first clinical trial in the world ever for this group of disorders. And as a part of that, we obtained skeletal muscle tissue, which we sent off to our collaborators at Columbia University for assessment of post-translational modifications, because although there are a wide range of different variants in this large gene, we believe that there may well be some sort of common pathomechanism in place, and we have some preliminary evidence which we're working on preparing now based off of that project. In terms of clinical trials, this was really my expertise coming in, and so this is where I felt I could really add to the team. This is the trial, which is a phase 2 trial of N-acetylcysteine, which is the precursor to the antioxidant glutathione. And this intervention was based on promising preclinical evidence in the zebrafish model, which I just showed you from a collaborator up in Canada. And we have recently completed this trial. Again, in red on the right-hand side, you have skills I've gained during my time here, in particular regulatory experience, which has been extremely beneficial for my career. So just to dip in with a little bit more detail on the trial, it was a parallel group, randomized, placebo-controlled, 2-arms trial, which was preceded by a 6-month natural history study where we focused on outcome measure development and validation. And the second phase of the study was again 6 months in duration, with individuals randomized to either N-acetylcysteine or placebo for a period of 6 months. And this was a really huge study. It was, as I mentioned, the first trial in this disease population. We had over 20 outcome measures. So this really was an incredible team effort and wouldn't have been possible without the support of our leadership. And formal results are pending, but we got the statistical results back last week, so I can dip into that as well. So in terms of the natural history phase, we've actually managed to recapitulate what's been shown in 3 independent preclinical models of the disease, which is that in this disease population, there is substantially high elevated oxidative stress as measured with urine isoprostane. In fact, 1.9-fold higher than what's expected in the general population. Participants only walked actually less than 80% of their predicted norms during the 6-minute walk test, which I'm sure many of you are familiar with. And we've corroborated the stable course of this disease, slowly progressive stable course of this disease. In terms of did NAC have an effect, results are pending. More to come on that. We actually have another clinical trial in the pipeline, which is a phase 1 trial. So that's actually focused more so on safety than efficacy. We are partnering with a pharmaceutical company to bring this to fruition at the clinical center. It's a small molecule, and will include around 10 participants focused on the pharmacokinetic, pharmacodynamic, and safety effects of the drug. However, we will include a few exploratory outcome measures which have been informed by Dr. Mayer's previous trial. So I've been very privileged to be able to present my work internationally and continue to do so through the support of the Intramural Program. And this has really been beneficial in terms of networking with world experts. As you know, I worked with elite level athletes, and this was really a new career step for me, and one that I'm very glad that I decided to take. So in terms of what next, I'm currently a visiting fellow, so I'd like to complete the phase 1 trial, which I just mentioned, as well as complete the Innovation Award Project preclinical phase with the zebrafish and cell culture models. And something which I feel I've had great support with here is in furthering my education. So I'm actually going to be obtaining my certification as a certified clinical research professional as part of my time here as a trainee. I would then like to move on to being a research fellow here at NIH and establish a longer-term natural history study for this disease. As I mentioned, the disease itself is slowly progressive, and so whilst the natural history phase of our trial that was recently completed was excellent in terms of being able to develop and validate outcome measures, to accurately assess the true natural history of the disease would require a more long-term study design. And of course, eventually, to become an independent investigator is where I'm hoping to end up. And I feel that my time here as a trainee has placed me well for that through the process of being able to establish reliable collaborations. So thank you to everybody that's been involved, and most of all, the study participants and their families. Thank you.